So in this video, I'm going to look at how the kidney works in terms of producing urine and how the water level of your blood, the water potential of your blood is controlled. So let's start just by thinking about the kidneys themselves. So if we think about the urinary system, which hopefully you are used to, you have two kidneys, one either side. They feed down into the ureters, which feed into the bladder. And then we have the urethra coming down to the outside of the body. And obviously feeding the kidney itself, you've got the renal artery, renal meaning kidney, carrying oxygenated blood, but also high in urea. And you've got the renal vein, which is coming away, carrying deoxygenated blood, but also lower in urea. So the renal artery, obviously it's oxygenated, but it's also got a high urea content, whereas the renal vein has a low urea content, because the idea is that our kidneys will filter our blood and remove the urea. The urea is a substance made in the liver from what we call deamination, which is the removal of the amine group from amino acids. So excess amino acids are converted into urea, and that urea is toxic. So it's then taken to the kidneys to be filtered out and removed in your urine. So your urine will contain urea, obviously water and ions, some salts. It shouldn't contain anything else at all. So let's have a look at the kidney itself. So the kidney, look at sort of a bigger version of it. Kidney itself is made up of different layers. So we have in the middle what we call the pelvis of the kidney. We then have these bits over here, which is called the medulla. And finally, we have the outer layer, which is called the cortex. Now, what I really want to look at are the tubules that are inside the kidney. Now, tubules or nephrons, as we're going to call them, they start up in the cortex, they dip down into the medulla, they come back up to the cortex, and then they finish up going down into the pelvis and out into the ureter, which is over here. Now, there may be several of these nephrons linking to one of the tubes that goes down. Okay, the tube that goes down is called the collecting duct, and we're going to label that in a minute on a bigger diagram. And you may have several different nephrons feeding into one collecting duct. So let's have a look at a nephron. So a nephron, starting at one end, will only deal with one nephron. Looks like this. Like so. So we already said before we labeled this, we said that was the collecting duct. Other bits that we need to know, this bit over here is called the PCT, and this bit over here is called the DCT. PCT stands for proximal, which is like front convoluted tubule. DCT stands for distal, i.e. the one further back, convoluted tubule. Convoluted just means bent, and it's a tubule. Down this big structure over here, this loop, is called the loop of Henle. And this capsule over here has two names. You could call it either Bowman's or renal capsule. And the idea is that the blood is going to enter here, form a knot of capillaries there, and that knot of capillaries has a name, which is the glomerulus. And then the blood vessels, the capillaries, drape themselves around the rest of the nephron. I'm not going to draw them all in, all the way around the rest of the nephron, all the way down to the collecting duct. So the basic principle is very simple. Okay, there are two main stages involved in the kidney. The first stage is the ultrafiltration, and we'll look at each of these in detail. Ultrafiltration, and that's the idea that you're going to remove everything that's small enough will go into the inside of the nephron. And then you're going to spend the rest of it selectively reabsorbing everything that you want back. So you'll spend the PCT reabsorbing, you'll spend the loop of Henle reabsorbing, you're going to spend the DCT and the collecting duct. Okay, so whereas ultrafiltration occurs in the Bowman's capsule, selective reabsorption occurs in the PCT, the loop of Henle, and the DCT and the collecting duct. So we need to look at each stage in turn. It seems a really odd way to do it, but it's how it works. Take everything you want into the filtrate and then reabsorb everything except the things you want to go out into the urea. So let's have a look at the process, first of all, of ultrafiltration, which, as we said, occurs in the Bowman's or renal capsule. So I'm going to just draw the picture of just the Bowman's capsule. So here is our capsule, like so. And remember we said that there was a knot of capillaries in here called the glomerulus. And that's where the process is going to take place. So the important thing to note is that there is a high hydrostatic pressure. That hydrostatic refers to water. So there's a high hydrostatic pressure in the Bowman's capsule. I'm just going to call it the BC for short, in the Bowman's capsule. And the idea is that this filters all small molecules into the nephron. The only thing it leaves behind in the blood are large things. So you've got the red blood cells, the white blood cells, platelets, and your large proteins. But we need to know a little bit more detail than that. So let's take a sort of cross section through this area. Okay, so what do we have? So if we think about what we have, we first of all have our capillary. Okay, and you should remember from the AS that capillary is made up of these endothelial cells, square ones, squamous, but we'll just call them endothelial cells. And then you have underneath that, that the endothelial cells are attached to is something called the basement membrane, which is a really fine filter. And then underneath that you have the capsule, but the capsule, the actual cells of the capsule are made up of these funny looking cells which have finger-like projections, and they are called podocytes. So we're going to take the substances from here through the gaps between the endothelial, through the basement membrane, through the pores in the podocytes, into the inside of the Bowman's capsule. That's the other side of the Bowman's capsule. So these small molecules, if we're describing it, we would say the small molecules, e.g. glucose, always good to give an example, but it could be glucose, amino acids, fatty acids, glycerol, ions, vitamins, water, urea, etc., are filtered through the gaps between the endothelial cells basement membrane, which as I said was the finest filter, and the pores in the podocytes. Now, how will you know when to answer this and when to just give a brief explanation? The answer is it depends on the number of marks in the question. If the number of marks in the question is just like a 2-mark question, they're really looking for the high pressure and the small molecules, e.g. glucose, leaving behind the large proteins. But if it's a 5-mark question, then they're looking for you to specifically name the pathway through. So you just need to just look at the question and how many marks it is in terms of the description of ultrafiltration. So if we go back to our Nephron, we've done the filtration process. So we now have in here glucose, amino acids, fatty acids, glycerol, vitamins, minerals, water, urea, you name it, we have it, with the exception of red blood cells, white blood cells, platelets, and hormones, which have been left behind. We now need to reabsorb all of the substances that we need. We're going to need to take back all of the glucose, all the amino acids, all of the fatty acids and glycerol, some of the water and some of the ions, and leave behind just urea, some water and some ions. And that's what the process of selective reabsorption is. So let's look at just a couple of aspects of it. So the first one is in the PCT. Now, we're not going to go through everything that we reabsorb, but the important things to say is that all of the glucose, amino acids are going to be reabsorbed. They're all going to be taken back, reabsorbed into the capillary from the PCT. All of them. Can't leave any of them behind. So if we go back to our nephron, here was our Bowman's capsule. We're just going to concentrate on the PCT here. Okay. Like that. Yep. So we now have say, we'll just deal with glucose. We have all the glucose that was in our bloodstream. Now, obviously, if you have too much glucose in your bloodstream because you're diabetic, you can have a problem reabsorbing it. But the idea is that all the way along the PCT, we're going to reabsorb the product so that by the time we get here, there is no more glucose left. And it's going to be reabsorbed by a mixture of active transport and facilitated diffusion. And this is very similar to what we covered in reabsorption of the small intestine, in the digestion. So if we've got our glomerulus there, we've got our capillary running around here. Again, we're going to just take a sort of cut through and see what is happening here. So these cells that are in here, these cells are those columnar epithelial cells. So we're going to draw one of those in. It's a good revision of this process. So we've got our columnar epithelial cells there, like so. Then we've got our blood, which is there. And we have the other side of the PCT, which I'm not going to draw in properly. So I'm only interested in this particular cell at the moment. So if you remember from small intestine absorption, there's going to be some different things that are going to be involved here. Okay, so we have a sodium potassium pump. We have a glucose channel for facilitated diffusion, and we have our glucose sodium co-transport protein. So our glucose is sitting here at the moment, and we've got to reabsorb it all the way back into the capillary, which is over here. So the first thing that's going to happen is that the sodium ions are going to be pumped out of the columnar epithelial cell by active transport. And that's important. That is going to create a concentration gradient. Create a concentration gradient, that's going to allow sodium to want to come in. So that's the stage number one. Second thing that's going to happen then is that the sodium ions are going to want to come in down their concentration gradient, and when they do that, they will pull with them glucose through that co-transport membrane. So the sodium ions down the concentration gradient co-transport membrane, co-transport protein, pulling glucose against its concentration gradient. So now we've got the glucose inside the cell, we now just have to get it outside. So all that will happen now is that that glucose will be quite happy to now move by facilitated diffusion into the capillary. Glucose will now move by facilitated diffusion down its concentration gradient into. So that process will allow the reabsorption of all of the glucose. Now, remember I said that if you were diabetic and you have too much blood glucose in your plasma, then you'll have a large amount of blood glucose in your filtrate. And what happens is you can't actually reabsorb it all, because all of the protein channels and protein carriers get saturated, and there's not enough protein channels and carriers to reabsorb that large level of glucose. So therefore glucose ends up still in the filtrate at the beginning of the loop of Henle, and therefore you end up with glucose in your urine. Right, I'm going to leave that video here. I'm going to pick up from the loop of Henle in the next video.